Tom, I can't see to admit people, just so you know.
Oh, good. Everyone's admitted, Margot, so I think we can start.
Amazing. Hi, everyone, and thank you so much for joining us today from wherever in the world you are. My name is Margot Monfarid, and I work for the International Coral Reef Initiative Secretariat, which is a global partnership for the preservation of coral reefs. It's my pleasure to welcome you to the First for Coral webinar series in partnership with the Reef Resilience Network and the NOAA Coral Reef Conservation Programme, hosted under the United States of America Secretariat. The topic for today is recent responsive events and the need for integrated response planning. So just before we start, I have some quick housekeeping to go through. So please remain muted throughout the call. Should you wish to speak, please click the raise the hand feature in the reaction section. We'll then unmute you and allow you to take the floor. Presenters will have control of their own screen. In the case of technical issues, please notify Tom Dallison or myself, and we have a backup copy. Presentations will be delivered in English with Spanish interpretation provided during the meeting. For the best quality audio experience, we recommend using a headset and a microphone if possible. The meeting is also being recorded, and we'll share this with you alongside the meeting presentations via the dedicated webpage on the ICRI forum. Lastly, we kindly encourage each participant to have their video on throughout the entire duration of the meeting, as this will enhance the overall meeting experience. So just to run through the agenda, we have a very rich and packed agenda, which will last about an hour. We'll have welcoming remarks from Michael Lamier, and then we'll move into our presentation topics, which will include stony coral tissue loss disease, invasive species, lionfish management in Mexico, the coral bleaching toolkit, the need for post-bleaching monitoring efforts focusing on the causation of coral mortality, the management actions taken in the US in response to the El Nino event in 2023, then a Q&A session where you're welcome to ask some questions to the presenters, and then we'll move back into concluding remarks. Unfortunately, due to a last-minute conflicting schedule, Jennifer Koss is not able to be with us today, but I am pleased to introduce you to Michael Lamier, the International Lead for NOAA's Coral Reef Conservation Programme. Mike, the floor is yours.
Great. Thank you so much, Margot. Yeah, good evening, good afternoon, good morning, depending on where you are in your present location. As Margot mentioned, I work for Jennifer Koss, the director of the United States National Oceanic Atmospheric Administration's Coral Reef Conservation Program. She does extend her apologies for not being able to attend. She's helping her mother recover from surgery this morning, but she's certainly here in spirit. All right. On behalf of our ICRI co-chairs, Jennifer and Christine Dawson, we welcome you all to the first #4Coral webinar series. For those of you who are new to ICRI, it's a global partnership between nations and organizations that strives to preserve coral reefs and related ecosystems. ICRI has 101 members, including 45 countries that are the custodians of 75% of the world's coral reefs. And this year's a special year for ICRI, celebrating its 30 years. Let those emoji confettis fly if you like. Also, stay tuned to ICRI's social media platforms where updates will be shared about this special 30-year milestone. Thank you all for taking the time to be here to listen, learn, and share your knowledge and experiences on response events and the need for integrated response planning. As we all know, threats to coral reefs do not recognize international maritime boundaries and require knowledge exchange and coordination to be effectively addressed. This collaboration exemplifies the importance of ICRI, its networks, and its members. Today's webinar is full of great presentations from our amazing partners who are well-versed in responding to disturbances. Their collective knowledge gained on this subject indicates response efforts often have common elements that can inform integration. Theme 3 of ICRI's plan of action calls for an integrated response framework, which should include the application of best practices from individual response efforts, restoration, and recovery actions to more effectively address the ever-evolving threats facing coral reef ecosystems. During this webinar, we'll first hear a variety of presentations on response efforts and plans, Please hold your questions and comments until after the last presenter. And lastly, as you listen to the presentations, please keep the integrated goal in mind, as your input and guidance on designing the integration process will be critical for success. So with that, thank you very much. And Margot, back over to you.
Thank you so much, Mike. Now I'm pleased to introduce you to Caroline McLaughlin, who serves as the National Coral Disease Coordinator with the Florida Sea Grant in partnership with NOAA's Coral Reef Conservation Program. She's going to tell you more about Stony coral tissue loss disease. So the floor is yours, Caroline.
Let me just share my screen. And then, sorry, 1 second, loading. Here we go. This should. Sorry, I don't know if you can still.
We don't see. We see your
okay,
notes.
Sorry. Can you hear me now? Okay, I'm sorry. It keeps saying Zoom has quit unexpectedly. So I don't know what's going on. Can you still hear?
We can hear you, but we don't see your screen anymore.
Okay, I apologize. I keep getting error messages saying that Zoom has quit. So let's one more try. Hopefully
you're
this works.
absolutely fine.
Okay, we're good? Can you see?
Yes, perfect. Thank you.
Okay, perfect. Apologies. So again, my name is Caroline McLaughlin. Thank you so much for having me. I'm the U.S. National Coral Disease Coordinator, and I work in a partnership position with NOAA's Coral Reef Conservation Program and Florida Sea Grant. So today, I'm going to talk to you about integrating response frameworks in the context of stony coral tissue loss disease. So I'll provide a very brief overview of the disease and then talk about how we've been approaching disease response at the national level in the United States. So just a very brief introduction to the disease. For those of you who are not familiar, stony coral tissue loss disease, or SCTLD, is arguably the deadliest coral disease that has ever been detected. One of our colleagues in Mexico really described it best, that it's likely to become the most lethal coral disease because of its very high prevalence, because of the high number of susceptible species that it affects, because of its transmissibility, and because of the high levels of mortality that are exhibited by corals that are affected by the disease. So as the name suggests, SCTLD is characterized by one or more lesions that may form anywhere in a coral colony. The lesions spread quickly and cause denuding or loss of tissue at a very fast rate. So the disease was first detected off of the coast of Florida back in 2014. It has since spread to 30 different countries and territories throughout the wider Caribbean. And so I think this gives you an idea of how widespread and pervasive this disease is. I will note that the disease has not yet been detected outside of this region. So it has not been detected in the Pacific or in any other area at this point in time. So we're still not really sure what causes the disease. Corals respond well to the application of an antibiotic paste. So it's thought that there is some bacterial component. However, there's another hypothesis that suggests that the disease may be caused by a virus that affects the zooxanthellae and not the corals themselves. So how is the disease spreading? We know that the disease can be spread via direct contact with infected corals, through water, and also that sediment can act as a reservoir. In terms of long-range transmission, the prevailing theory is that the disease is being spread by ships, specifically via ballast water or biofilms on a ship's hull. So the impacts have been dramatic, to say the least. Many places have seen an overall loss in coral cover just from this disease of around 50%. Some areas have seen a 90% decline in the highly susceptible species. In Florida, certain species like this pillar coral have now become functionally extinct. Conservatively, it's probably fair to say that about half of stony corals in the Caribbean have died as a result of this disease in the past 10 years. And of course, at this point, we don't really know the full scope of impacts. We don't know what the impacts are going to be on fisheries, on tourism, on cultural values, the ability of reefs to attenuate wave energy. But what we do know is that these losses are going to be incredibly significant. So moving now into response. So what does response consist of? First, surveillance. People are out, they're monitoring reefs, they're looking at disease status, and they're trying to evaluate the success of treatments. They're applying different treatments, with the most successful being the application of an antibiotic paste. Coral rescue and restoration have also been a really big focus, with the goal of preserving coral biodiversity to ultimately help restore reefs that have been heavily impacted by this disease. So thinking about response at the national level in the United States, in 2022, NOAA released an implementation plan for its SCTLD strategy that outlines a 5-year course of action with very specific activity plans laid out within the implementation plan. And it includes activities like research for new intervention or treatment techniques, increasing monitoring and surveillance, establishing land-based coral nurseries, and increasing engagement at the international level to help prevent disease transmission. So much of the work to coordinate the U.S. national response has been done under the umbrella of the U.S. Coral Reef Task Force, which has a working group focused on coral disease. So the U.S. Coral Reef Task Force is a body that brings together federal, state, and territorial agencies that are all focused on protecting coral reefs. Just this past October, at the task force meeting in the U.S. Virgin Islands, the task force unanimously passed a resolution focused on national action to combat the impacts of coral disease. So the 2 priorities highlighted in the resolution are preventing further coral disease transmission and also enhancing coral rescue and restoration in areas that have been heavily impacted by SCTLD. There's a lot of coordination that's happening at the international level in the Caribbean via the SCTLD Caribbean Cooperation Team, which NOAA helps to co-chair. And that team focuses on tracking the disease, sharing information, and also providing training, tools, and products to an international group throughout the Caribbean. So I just wanted to highlight that there are a number of Caribbean Cooperation Team members that are also ICRI members. So in terms of the Pacific, as I mentioned, SCTLD has not been detected in the Pacific, and we really don't know the likelihood of it making it out there. However, if the disease is being spread via ballast water and biofilms, it is possible, in theory, for the disease to make it through the Panama Canal and into the Pacific Basin. And so it's really, really important to be prepared and to continue to focus on preventing the transmission of the disease. So in terms of Pacific preparedness, we've been focusing on holding workshops that are focused on preparedness planning, surveillance, options for treatment, sampling for the disease, and also different ways to increase collaboration throughout the region. We're working to provide sampling kits to individual jurisdictions and are developing an intervention toolkit that will walk jurisdictions through the process of creating their own intervention and response plan. We have developed surveillance guidelines for use in the Pacific that include information about where you should be focusing surveillance and steps to take if you think that you see SCTLD in the water. So in the United States, a lot of the efforts in terms of preventing disease transmission are led by the Coast Guard and the Environmental Protection Agency. They have compiled and distributed ballast water best management practices to the shipping industry. The Coast Guard is working to identify high-risk ports and ships and to increase compliance checks. They are analyzing vessel movement to determine where ships are arriving in the Pacific that have recently visited the Caribbean in SCTLD-infected waters. And then they've developed an alert system for these vessels. And so if a vessel arrives in the Pacific from an area in the Caribbean that has been infected by SCTLD, they forward the ballast water management reports to the appropriate captain of the port zone. So thinking now about integrating response planning frameworks and why we might want to integrate SCTLD into broader response plans for other types of disturbances. So the state of Florida is a really useful case study. They have recently merged their SCTLD response team with another initiative to bring together activities that are focused on response to broader disturbances and also recovery from these disturbances. The framework has been used to mobilize rapid response to other emerging or existing threats. And then there's activities that are focused on reducing these threats and also enhancing ecosystem integrity. At the national level, we are right now expanding the scope of the Coral Disease Working Group to focus on other types of disturbances like invasive and nuisance species, bleaching, and major storm events. And by integrating response planning for all these types of disturbances, you're really increasing the efficiency of people's time and working together and kind of taking advantage of these existing frameworks so you can effectively respond to other issues as they arise. I will say we've developed a number of different resources specific to SCTLD that I would be happy to share with anyone. They're located on the NOAA website, And again, please feel free to reach out and I'd be happy to share those resources. And that's it. Thank you.
Amazing. Thank you so much, Caroline, for that very insightful presentation. It was great to learn more about your work on Skittledy. So now I am happy to introduce you to Maria del Carmen Garcia Rivas, who is a researcher and ranger with 25 years experience working with communities to protect wildlife and restore ecosystems. She was one of Mexico's first female instructors and amongst the first women to work with the country's fishermen and navy. Maria is going to tell you more about invasive species and lionfish management in Mexico. She will be presenting in Spanish, so if you do need to switch, please use the interpretation bar at the bottom of Zoom, and you can head to the English channel. The floor is yours, Maria.
Gracias. Gracias, Margot. Un gusto y agradecer de antemano pues la invitación. Es muy importante. Yo actualmente soy directora del Parque Nacional Arrecife de Puerto Morelos. He estado en diferentes áreas del Caribe mexicano. Pertenezco a la Secretaría de Medio Ambiente y Recursos Naturales. Entonces, les voy a presentar el programa de alerta temprana del pez león. Pues básicamente el pez león es una especie invasiva y hay que recordar que son las amenazas más importantes a la biodiversidad. Y esto se debe principalmente, es una especie que se usa para el acuarismo y llega a las aguas del Atlántico desde Florida, desde acuarios por diferentes liberaciones accidentales. Esto en el 2009, en el 1992, donde Florida pues informa que tiene esta amenaza. Es una especie que es muy fácil de colonizar debido a que es generalista, puede estar en el manglar, en el río, en los arrecifes, a 0 metros hasta 100 metros de 300 metros de profundidad, aguanta temperaturas de 10 grados y puede estar tanto en estructuras naturales como en estructuras artificiales, es decir, barcos, algunos muertos, algunos arrecifes artificiales. Por otro lado, pues es voraz, es un carnívoro que puede ingerir crustáceos en sus etapas juveniles y también peces, inclusive el propio pez león. Y por otro lado puede ampliar su estómago hasta 30 veces. Entonces, en su época de colonización, pues tiene todos estos atributos. En reproducción, igual que muchos peces, puede poner hasta 30,000 huevos en una fresa, esto cada mes. Y por otro lado, sus hábitos son activos generalmente en tiempos crepusculares o en la noche, lo cual hace difícil la captura. Y ya nuestros colegas de Estados Unidos identificaron que tiene un impacto importante en el arrecife, sobre todo en el reclutamiento de peces, disminuyendo el mismo. Y pues hay que recordar que las especies invasivas con algún tiempo pueden llegar a adaptarse siempre y cuando el ecosistema sea resiliente y esté en buenas condiciones. En México llegó en el 2009, primero el primer registro lo tenemos en la isla de Cozumel, que es la capital del buceo. Posteriormente, pues ya bajó por toda la costa de Quintana Roo, que es el Caribe mexicano, donde tenemos poblaciones humanas en el norte, principalmente por buzos y algunas cooperativas pesqueras, en el sur menos poblado, pero sí con ojos en el agua. Entonces ya para 2010 identificábamos en dónde estaba distribuido, que era prácticamente en todo el Caribe mexicano, inclusive ya se fue al Golfo de México, a las aguas de Veracruz. Dada esta circunstancia, nos reunimos con nuestros colegas de la NOAA que ya tenían una experiencia, NOAA y RIF, y quisimos hacer de esta amenaza una oportunidad. Es un pez, el cual puede consumirse, el problema es que tiene espinas que son letales, y hicimos un programa de maestro con 6 componentes, los cuales los voy a mencionar, alineado también a nuestros programas de especies invasivas de la CONAVIO. Pues básicamente el primer elemento, que es la comunicación, lo dirigimos tanto a las personas de a pie para que conocieran el problema y consumieran el pez león, y a los pescadores y prestadores de servicio, a la gente que está en el agua, para que nos avisara en dónde está y poder entonces actuar con el problema, que es capturarlo. En relación a la captura, pues a los pescadores no les tuvimos que enseñar a pescar, sino más bien cómo manejar la especie con estas espinas venenosas. Y a los prestadores de servicio, pues sí, una clase sobre pues cómo manejar hawaianas sin dañar el arrecife, porque no cualquier persona sabe capturar. Entonces, pues hubo esta parte de capacitación. En relación al monitoreo, los estudios que se desarrollaron fueron básicamente para ver cómo colonizaba esta especie, cómo iba llegando a las aguas mexicanas, qué presas contenía. Por otro lado, también pues promover fondos para seguir investigando y también pues qué necesidades de estudios para su mejor manejo como parte del comportamiento para optimizar las capturas. En la parte del marco legal, si bien los pescadores tienen permiso de pesca, los pescadores comerciales, las cooperativas, los prestadores de servicio turístico no, entonces hicimos un acuerdo con la Secretaría de Pesca para que ellos lo pudieran hacer. Por otro lado, también se dieron facilidades para las autorizaciones de investigación de esta especie, así como pues los impactos en el arrecife y algunos acuerdos en los programas de subsidio mexicanos para que se incorporara en estos subsidios el monitoreo, captura y comercialización del pez león. En relación al mercadeo, pues lo que hicimos básicamente fue promover el consumo de esta carne. Se hicieron estudios de nutrición, lo cual pues salió como una carne muy nutritiva, planes de negocio para los pescadores para que vendieran el producto adecuadamente, identificar compradores, buscarles créditos para que lo pudieran posicionar en el mercado y pues también el mercado de artesanías del cual vamos a hablar. En relación a las pesquerías, aquí tenemos los resultados de las dos cooperativas campeonas, que fue la cooperativa de Cozumel, que intercedió tanto en Cozumel como en Siancán, y la cooperativa de Puerto Morelos. En 3 años sacaron más de 121 organismos, casi 15 toneladas de filete, lo cual equivale más o menos a $306,500 en estos 3 años. En relación a las artesanías, aquí las tenemos y de hecho las traigo puestas. Es un pez muy bello, las aletas no dejan de serlo, entonces se capacitó a las esposas de los pescadores. Esta fue una actividad totalmente innovadora, el ya incluir a las mujeres en esta comercialización, en esta también hacer estas artesanías, esta actividad, estas manualidades, y bueno, pues ellas hasta la fecha siguen vendiendo estas hermosas artesanías de pez león, tanto en Xcalac como en Isla Mujeres. En nivel internacional, uno fue a nivel regional, en el sistema arrecifal mesoamericano, en el monitoreo de AGRA incorporamos la especie de pez león, hemos tenido foros en el ICRI, en el Gulf and Caribe Fisheries Institute, en foros temáticos, intercambios de experiencia para ver las partes de mercadeo, captura, comercialización y participar en diferentes redes, como ya bien nos lo decía Dave al inicio de este foro. ¿Cómo está ahora el pez león en nuestras aguas mexicanas? Bueno, hemos hecho el monitoreo, este programa continúa. En el caso de Puerto Morelos, que es la gráfica azul, lo que vemos es que en estos últimos años del 2018 para el 2023 hay una densidad muy baja de peces león, de 12 y ha bajado hasta 3, siendo que cuando inició la problemática llegábamos a tener en Puerto Morelos hasta 70 peces león por hectárea. En Xcalac disminuyó, tenemos datos del 2019, pero pues es una tendencia. Si bien el impacto ya se había identificado, hicimos estudios específicos en México, en el caso de la pesquería de langosta espinosa no afectó, entonces también hubo huracanes durante esta historia del pez león, entonces pues no hemos podido identificar con precisión, ya que es multifactorial, la salud del arrecife tiene que ver con otros factores. Conclusión, este fue un programa exitoso. El pez león se registra como una especie invasiva, se hizo a tiempo y los factores de éxito fueron principalmente encontrar a un líder que pudiese llevar este programa, que fue la CONAM, que tenemos representadas áreas naturales protegidas en toda la costa del Caribe mexicano. Por otro lado, socializar a tiempo, tener un marco legal adecuado, una solución con un financiamiento, que fue este consumo, y por otro lado, un sistema adaptativo y la continuidad del programa. Y nunca quitar de vista que es una especie invasiva, que si bien la comercialización ha sido un éxito, no queremos que se quede en nuestras aguas mexicanas ni en nuestras aguas del Atlántico. Pues por mi parte sería todo. Muchas gracias.
Thank you so much, Maria. That was super insightful and great to learn more about the international collaboration that you have going on as well. Now I'm pleased to introduce you to Andrea, who is the Coral Reef Alliance's Project and Outreach Manager for the Global Conservation Science Programme. She works in multiple groups worldwide to monitor coral bleaching. She also collaborates with conservation scientists globally to improve the Allen Coral Atlas bleaching detection features. So over to you, Andrea, and the bleaching toolkit.
Thank you, Margot, and thank you everybody for being here today. I'm delighted to share with you a little bit more about the Coral Bleaching Toolkit and Comprehensive Guide. So the goal of this toolkit is to gather a wealth of information, also actionable guidelines from the world and resources for understanding and monitoring coral bleaching events. 2023 was a devastating year for coral reefs, and I think many areas in the Caribbean saw firsthand the record temperatures that drive healthy coral reefs to undergo coral bleaching and then coral mortality. And this prompted the need to consolidate efforts to help teams on the ground respond to bleaching events by providing by different locations what are the efforts that are undergoing in those areas to find easily more information on how to monitor and respond to coral bleaching events. So here's a QR code if you want to check it out. I will talk a bit about what the toolkit includes. It has a summary of a brief introduction. It contains an overview of coral bleaching, and then it goes into detail about different tools that are being used to remote sensing tools that are being used to monitor coral bleaching, also on-site monitoring tools. It also provides a checklist on how to mobilize and protect coral reefs. And then there's a section about the future of coral reefs and other ways that you can help. So we'll start with the remote sensing tools. Briefly, there's different levels, the global and regional tools. We'll hear more about the NOAA Coral Reef Watch tool from our next speaker, Derek Mancello. We have the Allen Coral Atlas tool as well, Aqualink. At the regional level, there's CIMAR, which is basically for the Caribbean, the Coastal Marine Information and Analysis System. There's also the Western Indian Ocean and the Hawaii Pacific Island region climate tool. So once you go to the site, you'll hear and you'll see the this is an image from Mari Carmen. And you can start checking the different tools. There's a brief description. Here's the Allen Coral Atlas, the CIMAR, and then you can click on whichever one you're interested in depending on your location. When we go to the on-site monitoring tools and platforms, there's different levels of tools being used around the globe in the Atlantic, in the Pacific, and East Asia, and Indian Ocean. There's also new information coming in from the Red Sea, Persian Gulf, and Brazil. And once you go to the site, you can check which are the different organizations and tools being implemented in each of the areas. So if you go to an area that you work with and you don't see your organization that has a program for coral bleaching, please reach out to us so that we can also include it in the next update. And finally, there's also a checklist. So the goal of this checklist was to provide information for managers, but mostly for dive operators. This was inspired by the NOAA Coral Reef Information System, the NOAA CORS. And we wanted to summarize a checklist guide on what to do before, during, and after event, as I mentioned, geared towards dive operators and managers. So please head to the website either by scanning the QR code or by going to coralbleaching.com, and hope this is going to be useful for the location that you're working. Thank you.
Amazing. Thank you so much, Andrea. It's great to learn more about the fact that you've got new information on the Red Sea. And also I find your checklist super interesting and great to use as well. So thank you very much for that. Next, we have Derek Manzello, who is the coordinator of NOAA's Coral Reef Watch Programme, an effort focused on the monitoring of coral reef ecosystems through satellite and in situ observations. Derek will be addressing the need for post-bleaching monitoring efforts. So over to you, Derek.
Cool. Let me just share my screen here. All right. Can y'all see what's going on here? All right. So thank you very much. My name is Derek Manzella. I'm the coordinator of NOAA's Coral Reef Watch. As many of you are probably aware, we are in the midst of a large-scale ongoing mass coral bleaching event that's started about a year ago, actually. And this shows you the current forecast for bleaching in the Southern Hemisphere. And unfortunately, it's looking like the majority of reef regions in the Southern Hemisphere are at risk for bleaching this year. I wanted to point out that we had to modify our bleaching alert area levels at Coral Reef Watch. So historically, there were two bleaching alert levels, bleaching alert level 1 and bleaching alert level 2. Simply put, alert level 1 is when bleaching is expected. Alert level 2 is when we expect mortality to begin. So the issue became last year by October, the entirety of the Northwest Atlantic Ocean and the Caribbean Sea, including many areas of the Eastern Tropical Pacific, had greatly exceeded the definition of what we call an alert level 2. So we had to add in 3 new alert levels to better reflect the amount of heat stress that was being experienced. Again, along with these alert levels, we provide some context for the potential risk of ecological impacts. And these are based on the literature for what's happened in the past at these various alert levels. So as you can see, the majority of the wider Caribbean in the summer of 2023 experienced what we call an alert level 4. Now, an alert level 4 is 2 times the amount of heat stress that is an alert level 2. So at an alert level 4, we expect severe multi-species mortality and risk of more than 50% of the corals dying. Alert level 4 is the worst condition. This is analogous to a category 5 hurricane or cyclone. And there's been 3 examples of reefs that have experienced this in the past. And in those cases, mortality ranged from 89 to 99%. So we are conservatively estimating that at an alert level 5 condition, more than 80% of the corals are at risk of mortality. So the 2023-2024 mass coral bleaching event, it hasn't yet gone global simply because we have not yet received reports of significant widespread bleaching in the Indian Ocean. But it's essentially, we have severe bleaching being reported in all other ocean basins as of right now. And over the past 12 months, we've received reports of bleaching in at least 41 countries and territories across 5 different oceans and seas. So post-bleaching monitoring, a significant fraction of the mortality from a bleaching event might actually not be driven by the heat stress. And it's really important that everyone understand this because this provides an opportunity for us to save more corals. And the more we can learn for how corals respond to heat stress, the more we can intervene in the future. So a great example is what happened in 2005 in the US Virgin Islands. So the US Virgin Islands experienced what we are now calling alert level 4 conditions in 2005. Now, what ended up happening is many of the corals actually survived the heat stress and didn't directly die from the bleaching. But what happened was in the following 1 to 2 years after the heat stress event, there were multiple outbreaks of coral disease. So different types of coral diseases experienced outbreaks, and this led to a 60% decline in coral cover. So this is a very great example showing you that the corals themselves didn't actually die from the heat, but it was actually disease that came on after the heat. So this provides opportunities to do interventions for treating coral diseases. And this is just a hypothetical example of what could potentially occur following a heat stress event. So up there on the top is the heat stress that actually occurred in Florida last year. And down below are theoretical responses of different species. So we already know Acropora can drop dead quickly, so these require rapid intervention. So as soon as this heat stress starts, Acropora intervention plans need to be put in place. However, many of the other species, particularly in the Caribbean, have very different responses. So we know black band disease is stimulated by high temperatures, but stony coral tissue loss disease actually essentially goes away when there's heat stress, which is a very interesting result. So if we know more and more about diseases that are impacting corals after the bleaching event, that gives us an intervention timeline for responding to these events in the future. So in 2005, the main disease that came in after that bleaching event was white plague. We have a new coral disease in the Caribbean, as Caroline just pointed out, stony coral tissue loss disease. So if we have this kind of information like you see below, this would provide us, again, with an intervention timeline to know, okay, these species of corals will likely survive this heat stress. Therefore, we can focus all our attention on Acropora at first, but then we need to go out and intervene after the heat stress event for other coral species like the Orbicellas, Siderastria, Copophilia, Diploria, Pseudodiploria, all those other guys. And the other thing that happens quite a bit after bleaching is increased corallivory, which is basically the fancy way of saying coral predators can focus in on and really devastate survivors of bleaching events. So just to give you an example, in the Indo-Pacific, Andy Bruckner et al. Observed Drupella snails forming aggregation around corals that specifically resisted bleaching. So these were the bleaching resilient corals they observed, but then after the bleaching event, these aggregations of these snails came in and started feeding on them and causing mortality. Same thing in the Caribbean with Corallophilia that specifically target Acropora colonies with damage or disease. So essentially what you have is when you have a stressed coral, you can have these snails specifically aggregating onto damaged or diseased corals. And this is a huge problem in the Caribbean because things like Acropora are at such low abundances now that you'll get multiple snail individuals fixating in on that one patch of remaining live tissue, and they can basically eliminate and decimate tissue very, very fast. So again, removing corallivores is a well-known viable intervention to save corals from bleaching. And we just need to keep that in mind during the bleaching event. Just because your reef bleaches doesn't mean it's automatically going to die. There are other things that may happen after the bleaching that can be potentially addressed. So final thoughts: to save our bleaching survivors, we first have to understand the timing of subsequent disease and corallivore outbreaks. So are these events predictable in time, or do they happen kind of haphazardly and not by any kind of clock? If they are predictable in time and follow certain patterns, then that gives us a playbook for what to do next time it bleaches. And again, we need as much monitoring data as possible during bleaching, but for upwards of 2 years after the bleaching stops. And this is because, as I just said, these secondary stressors sometimes can take years to impact the corals. So corals can become immunocompromised and suffer disease upwards of 2 years after they recover from the heat stress. So it's important to have eyes on the reef for upwards of 2 years after a bleaching event. And this will help us identify resilient reefs, species, genotypes, and provide that blueprint for how to intervene and save corals during the next bleaching event. And just finally, I want to point out that preventing a local extinction of a coral species like Acropora palmata or Acropora cervicornis in the Caribbean could be as simple as picking snails off recovering corals. So thank you very much. I'd like to thank the Coral Reef Watch team for all the great work they do, and thanks for having me.
Thank you so much, Derek. That was super interesting, particularly the intervention timelines. And I myself know how annoying it can be to pick off Drupella off aquapora species and things like that. So thank you very much for your presentation. I'd like to move on to our last speaker, Jennifer Moore, who has worked on the conservation of Caribbean corals her entire career since joining NOAA in 2003, and listed the first ever corals as threatened under the Endangered Species Act in 2006. Most recently, Jennifer has been coordinating responses to the several disasters affecting Caribbean corals, and Jennifer will be addressing the management actions taken in the US in response to the 2023 El Niño event. The floor is yours, Jennifer.
Thank you very much. Share my. All right, is everybody seeing that okay? Okay. Well, thank you very much. I'm going to be talking primarily about our management response actions in Florida because of my co-lead, because I'm the co-lead of Mission Iconic Reefs, which is the largest U.S. coral restoration project that is taking place in the Florida Keys National Marine Sanctuary. I want to say right off the bat, these management actions, these response activities that we were able to undertake were really possible because of the foundation of having that extremely strong network of collaborators working on a coordinated restoration activity. So right as we began understanding that this was going to be an extremely hot summer last year, in about June or so, a lot of our restoration practitioners voluntarily stopped outplanting corals, knowing that those corals are already undergoing a moderate amount of stress from transport from nursery to the reef, and that stress would obviously be exacerbated by the increased temperatures. They voluntarily started doing that. And then the state of Florida and the Florida Keys National Marine Sanctuary created a moratorium on outplanting once the temperatures had consistently reached over 30 degrees Celsius. And then as Derek really set me up really well for this talk, as we all know, the acroporids in the Caribbean are doing extremely poorly. That is why they are on the US endangered species list and also on the IUCN red list. And in Florida in particular, we had fewer than 150 extant unique genotypes of Acropora palmata and about 300 of Acropora cervicornis. And so it was imperative that we try to save and rescue as many of those genotypes as possible so that we don't lose that genetic diversity and are able to then rebuild that population hopefully in the future. So the first activity to do that was a gene banking effort. We have two facilities, Mote Marine Laboratory in Sarasota, Florida, and the Reef Institute in West Palm Beach, which are about 300-ish miles away from each other, to provide two land-based, stable, distant facilities to hold these small fragments of the two species in perpetuity. These are meant to stay there forever, so we have a long-standing gene bank of these colonies so that in the future we can use them to continue to do restoration efforts should everything in the field die, which unfortunately we got very close to that this year. Another activity that our restoration practitioners undertook was not only to do genetic preservation, but to actually do nursery stock salvage. They evacuated corals both to on-land nurseries and deep water nurseries. And I will say that there was varying success in these activities. The acroporids are particularly sensitive to being cared for on land. They need, not surprisingly, high water flow, given the habitats that they grow in naturally, and a lot of light. And most of the conditions that people care for corals on land, they actually try to keep the light relatively low to prevent algal growth in the tanks. And so there was sort of a balance there. Plus, also, when the corals came in, the acroporids were already stressed. And so, unfortunately, most of those corals that were transferred, evacuated to land-based facilities in the acroporids did not fare very well. In contrast, the boulder corals that were evacuated did extremely well, and actually they started growing while they were in the land-based human care. And so we actually returned more colonies. We were able to do some fragmentation back to the water when the water's cooled to put back into the nurseries. We did have a lot more success with the acroporids by moving them to deeper water nurseries. The shallow water nurseries are typically in 20, 25 feet of water, and these deep water nurseries were established in about 70 feet of water. And so those corals were, obviously had less light and slightly cooler temperatures. And we found that the light combination with the temperature is really indicative of how severe bleaching is going to be. For example, we had many colonies, both in nurseries and observed out in the wild, that initially had bleached on their top surfaces, but if you looked underneath, they still had zooxanthellae. And so by moving these corals to deeper water, we both reduced the amount of light that they were receiving and also had them in slightly cooler temperatures. However, because we had so long of a duration, 20 or so or more degree heating weeks of these temperature increases, even down to 70 degrees, we did see the bleaching temperature threshold exceeded, but just not for as long as in the shallower water. There is another potential problem, though, with moving these corals to deeper water. Because of that lower light reduction, the longer they stayed in those deeper waters, they actually started showing signs of poor health because they were not receiving as much sunlight to photosynthesize and receive their nutrition. And so there's a balance, and you really need to be able to move those corals back into the higher light regimes once the temperatures have stabilized. Another thing that we're thinking about improving upon this for next year is being able to raise those coral trees up and down in the water column so that you can move them ahead of the corals becoming stressed into those deep water areas, but have them up closer to the surface while they're receiving and be able to receive the amount of light they need. But then as soon as temperatures start to indicate that they're going to be stressed, you can move them down and balance those two conditions. Another thing that we really ramped up was engaging in various different citizen training programs to help us do observations of where coral bleaching was occurring. We have within Mission Iconic Reefs an Iconic Reefs Guardian program that is able to get recreational divers involved in various aspects of the coral restoration pipeline, observing nurseries, helping to maintain nurseries, and eventually conducting site maintenance. But we pivoted, and to use that, again, existing foundation, we pivoted that training program to do bleaching observations in collaboration with other established networks in Florida to do bleaching observations. And again, Derek set me up very, very well. It was imperative for us to do monitoring. So we have done 2 synoptic monitoring cruises where we visited all 7 of the iconic reef sites, one in August of 2023 during not even quite the hottest or the height of the bleaching event, and then in just this past February. And we saw some differences. Obviously, a lot of the corals were bleached but still alive during the bleaching event in August when we did our cruise. But the majority of the outplanted corals in the acroporids did not survive when we did our follow-up monitoring in February. There were very few sites that still hosted Acropora palmata. Cervicornis did a little bit better, but our boulder corals fared much better. So even though we observed bleached corals in August, many of them had recovered when we revisited those sites in February. All of the results are not in yet, but we will be using this information to help us not only understand regional and site-specific responses to the bleaching event, but also genotypic response, because we do have some information about the genotype of the corals that were outplanted. And so therefore, we can then use that information to better inform activities going into the future regarding restoration of these species. Another monitoring technique is that we were using some novel ways to monitor during the event. This is a drone flown image where we see one of our reef sites in May had lots of color, and then during the height of the event, You could see the bleached acroporids in this image. There have been follow-up images also flown, but not processed yet. And so we will then have the ability to compare throughout the entire sort of year-long sort of monitoring of this particular site. It's hard to tell from these images, but you can zoom in quite close down to the centimeter level and really look at individual coral responses to the bleaching. And then from a management standpoint, we have to continuously adaptively manage. We will be doing that specifically for the revisiting the plan for the mission iconic reefs. We had a workshop in January where we brought together all of the implementation practitioners and managers to kind of do a retrospective on all of these interventions that we tried, plan for this year so that we can get ahead of it. We did get caught a little bit flat on our feet, even though we knew it was predicted to be a pretty severe bleaching year. And so we are prioritizing interventions. Other things that have been tried are individual shading of trees, and that seemed to also be very successful. So our practitioners are investing in developing those for their nurseries. Again, deep water nurseries, hoping not to have to do large scale evacuations because that provides a whole nother level of logistics and stress to the corals. And then moving forward, we want to incorporate this information on how best do we do restoration in the future. It seems pretty dire that we lost as much as we did of our restored acroporids. However, there are survivors and we need to learn from them and be more strategic in the future to make sure that we are going to be outplanting things that have the highest probability of success. One thing I didn't include in this slide deck was actually, again, Derek set me up very well, is we did do some targeted large scale removal of the corallivores, the snails in one of our sites in Carey's Fort Reef. And we picked that site because it had a little bit lower heat stress. And so there was still living acroporids there at the time. I don't have the results of that yet, so that's why I didn't share it. But we know that we made an impact because we were able to collect thousands of snails. And so, and we could see them preferentially marching towards the surviving acroporids. So we know that we did assist in survivorship at that site. We just I don't have all the numbers yet, so I didn't want to put that slide up. But I just wanted to thank all of the partners that participate in Mission Iconic Reefs. Again, all of these various activities that we undertook were Herculean. They were happened extremely fast in an extremely collaborated way. And that would not have been possible had we not already had this established network working together on Mission Iconic Reefs. So thank you very much for your attention.
Thank you so much, Jennifer. It was great to learn more about your adaptive management and also about your nursery stock salvage efforts. I particularly like your idea to raise the trees up and down as well. So that's super interesting. Thank you. Next, we have about 5 minutes for some Q&A. So if you would like to ask a question to our speakers, please raise your hand and we will unmute you. No questions at all? Please feel free. I don't think anyone will be scared to answer anything.
Margot, I can, oh, somebody's got their hand up. You can take that first. I got one later.
Hello. Hear me okay?
Yeah, we hear you very well.
All right, great. I'm Anila Quilmot here from Aracabessa, Jamaica. I was just wondering with the presentation where the corals were brought on land, if the water that they're in on land, was it water from the sea? Was it a circulating seawater? Was it a closed system? And then, if it's circulating water from the sea, is there anything in place to try to control the temperature of that water?
Yeah, I actually just responded in the chat, but yes, we had actually both types of systems, open and closed systems, but all of them have temperature regulation on them. So we were keeping them well below bleaching thresholds when they're in the land-based facilities.
Okay, could you explain like how that works? How the if you can, like especially for the open water system, because that's something we had considered, simple enough, it seems, to set up, but then that was our concern. How do we actually control the temperature?
Yeah, there's large scale chillers and heaters to maintain the water temperature, because we also in Florida get cold water in the winter. And so those systems that have corals in them year round also need to sometimes be heated to keep them sort of in optimal conditions. So they're chillers and heaters that work on the incoming seawater before it enters into the actual raceway tanks that hold the corals.
Thank you.
Thank you so much for that question. Does anyone else have any more questions?
I have a question. Can you hear me?
Yeah, go ahead.
So Belinda Morrow from White River Fish Sanctuary, Otrius, Jamaica. So would you say that with the acropora corals that you were trying to keep alive there, would you say that it was more successful to try and keep them alive in water nurseries, I mean in situ or ex situ? Because I had thought that you had said that your efforts this summer with the acropora had not been that successful, and you'd lost a lot. So is it just that you lost a lot because by the time you started doing it, they were already stressed? Or is it that you really think that they perhaps just don't do so well in the lab situation, and one would be better to just try and really keep them alive in situ?
So there's a lot of nuances in that. So in terms of the gene banking, the small sized fragments that we moved into land-based facilities, those fared very well. We've lost very little of those. The ones that were evacuated into various different land-based tanks, they were shipped in some cases hundreds of miles away. Those did not fare as well in the land-based. And we think it's multiple factors. We think it's because they were already stressed when they came in. And then it takes a lot of human care to care for these corals, because they evacuated thousands, up to 12,000 corals. So having that many corals in human care is just very challenging, particularly the acroporids, because of how much light and water flow they need. And I think in some of the systems, they weren't set up initially to actually provide those conditions. So I think the corals that came in stressed were then further stressed because they weren't receiving the amount of flow that they really need or the amount of light they need to be able to photosynthesize and help them recover. So I think those were sort of like multiple factors that contributed to why they didn't do as well on land. And then there's also just a lot of logistics and transport stress. And so if you can move them into deep water nurseries in situ where you can provide lower light and lower temperature, that is what the majority of our practitioners are going to be focusing on for this year, rather than the large scale evacuation of the acroporids to land-based facilities. However, gene banking, they intend to try to have as much as they can in land-based facilities, not only in our centralized gene banks, but a lot of our practitioners want to set up land-based facilities for their own internal gene banking so that they have a copy of basically every coral on land, and then work with some of these deep water nurseries to for their stock. And the boulder corals fared much better on land. And so depending on how bad the bleaching event is meant to be, how many deep water nurseries they're able to establish, they may also evacuate the boulder corals, the star corals, brain corals, etc., to land.
Thank you.
Thank you. And I saw another question in the chat that I'll just answer real quickly. Yes, we know that corals that would have died in the wild that were evacuated to land and saved may not be the best representatives of ones that will continue to be able to thrive into the future. However, what the primary focus is in population management is to not lose any genetic diversity, because we can reproduce these corals sexually. And we actually have some anecdotal evidence that some of our sexual recruits did survive the bleaching. So we don't want to lose any of the genetic diversity of these species. And that's why it's really important to evacuate them, gene bank them, do as much as we can to save them, so that we have not lost any of the genetics that could potentially provide the resilience going into the future.
Thank you so much, Jennifer. That was a great response. I'm just going to jump in there and pass the floor over to Jen. I see her hand up, but unfortunately we don't have time for more questions after that. So I will then pass over to Michael for the closing remarks. If you do have questions, please feel free to email those in. So over to you, Jen.
Good morning. I was able to jump on for a little while. Looks like we're having a good response here. Of the panel speakers, thank you. All of your talks were really good. We are thinking about here in NOAA as we rewrite our strategic plan to kind of merge disturbance response into a single line of thought with strategies and objectives associated with that. And I'm wondering if you all have thought about what does integrated response planning and integrated response plans look like? Because there seems to be sort of a commonality of no matter what the disturbance is, there may be the need to actually directly intervene with corals, rescue them, move them. And as a U.S. Focus, we're definitely going to try to do it in the NOAA strategic plan, which will become a national level plan, and it may have implications internationally. So if Jen, maybe you've thought about more holistic planning and best management documents that we can put together so that people aren't having to go, oh, I have a bleaching event. Oh, I have a disease. I'm jumping all over the place to get the right course of action to take.
Yes, we haven't really talked about a unified single one, but most of our practitioners are trying to develop their own emergency response plans, and it probably would be great for us to roll that up. I mean, we've kind of talked about it for mission iconic reefs, but I think trying to roll it up to a higher level would be really, really great for the community.
Any other panel members? Okay, back to you, Margot.
Thank you so much, Jennifer. I'm now going to pass back over to Mike Lamier for some concluding remarks.
Okay. Well, great. Thanks, Margot. Yeah, it looks like we had a great turnout. Looks like I think our participants peaked at about 63 participants, which is great. So I just tried to take some notes and write down some common integration best practices I heard. So merging teams, engaging citizens, rapid information sharing, leveraging extensive networks of experts, utilizing online platforms for information sharing, and monitoring and adapting all seem to be like common best practices. Yeah, so thanks again to Caroline, Maria, Andrea, Derek, and Jennifer, and all the participants for joining today. Thanks again to Margot, Tom, and Francis for organizing and leading us today. Keep an eye out for future #4Coral webinars in the near future. Enjoy the rest of your day or evening. Thank you.
Thank you so much, Mike. And with that, if you do need more information, this will all be shared via email after the webinar, and it will include a link for the recording, presentations, additional resources. They can also be found on the dedicated ICRI webpage. The next webinar will take place on the 4th of April at 9 o'clock British Summer Time, so please join us for that. Lastly, a big thank you to all the speakers for your fabulous presentations, and to our partners of the webinar, the Reef Resilience Network and NOAA Coral Reef Conservation Programme, hosted under the ICRI chairmanship of the USA, as well as our financial supporters, the Principality of Monaco and the Government of Sweden. And also thank you to all of you for taking the time to attending this webinar, and I look forward to seeing you all at the next one.
Thank you. Have a great day.
Thank you. Gracias.
Thank you, everybody. Goodbye.
Thank you.
Bye.
Thank you.